Alright, today's video discussion is on protein synthesis. When we talk about protein synthesis, we're referring to the process which starts with a DNA molecule and ends with a protein. This is an important process that occurs throughout the body because we need proteins for a variety of functions in the body. Major function of proteins are enzymes, acting as enzymes to speed metabolic reactions. So without protein synthesis, we would not have any functional enzymes. So very, very important process. So we're going to talk about the different steps going from a DNA molecule ending with a protein, and those steps are DNA replication, which is simply making another copy of DNA, transcription, when we're taking the DNA information and converting it to mRNA or messenger RNA, and translation is when we're taking the information from messenger RNA and linking it to different amino acids, eventually forming a protein. So when you look at DNA, where we find DNA is in the cytoplasm, or I'm sorry, in the nucleus of cells. It's stuck in the nucleus. It cannot leave the nucleus. So therefore, the process of transcription and translation requires messenger RNA to occur because messenger RNA is found in the cytoplasm and can travel into the nucleus. We'll talk about that in a little bit more detail. But looking at basic nucleic acid structure, specifically DNA, is made up of three major components. We have a phosphate group here along the side, and we have the sugar, which is deoxyribose, and that forms the sides of the DNA ladder. So if you picture DNA like a simple ladder that you would climb on to reach something tall in your house, that's what DNA looks like. The sides of the ladder are repeating units of deoxyribose sugar, phosphate, deoxyribose, phosphate, and so on. The rungs of the DNA ladder are made up of 4 different nitrogen bases. We have guanine, cytosine, thymine, and adenine. Those are the 4 different nitrogen bases. If we look at the nitrogen bases, based on their molecular structure, we find that thymine can only bond to adenine, and guanine can only bond to cytosine, and vice versa. Cytosine and guanine are always a pair, and thymine and adenine are always a pair. We never will see guanine and adenine bonded together, nor will we see cytosine and thymine bonded together. So based on their molecular structure, that's what holds the rungs of the DNA ladder together, are the bonds between these nitrogen bases. So that's what these lines are representing, are the bonds between the nitrogen bases, and specifically the type of bonds we talked about in the chemistry of life. There's covalent bonds, ionic bonds, and hydrogen bonds. Remember that hydrogen bonds are the weakest of bonds, and that's what holds the rungs of the DNA ladder together. That's what bonds these nitrogen bases together, are the hydrogen bonds. So although they're weak, they do play a very important role in DNA structure. So these hydrogen bonds are broken when we need to make a copy of DNA, which we'll talk about in just a little bit. So know that these are hydrogen bonds holding the rungs of the DNA ladder together and between these nitrogen bases. So in the process of DNA replication, essentially what's happening is we're taking this DNA molecule that is a ladder, but essentially a twisted ladder. So it has this appearance here. So the sides here, this is where the sugars and the phosphate molecules would be found. And these nitrogen bases in the middle, remember, are forming the rungs of the DNA ladder. So this molecule is twisted into a helix shape. So we call DNA a double helix because of the twisted shape and the 2 sides that form the DNA molecule. So when we want to make another copy of DNA, for example, when a cell is getting ready to divide and make a copy of itself, that's called DNA replication. So to replicate something means to copy it. So what happens is DNA will separate or unwind with the help of an enzyme, which is called DNA helicase, and that will unwind the DNA ladder, and then another enzyme will come in and start dropping new pieces of DNA onto this unwound DNA ladder, and that's with the help of another enzyme called DNA polymerase. So we'll watch this animation here to get a little visual picture of what exactly DNA replication entails. So I'm going to turn the audio
DNA
off
replication.
here in this case. So if we look at this wound DNA molecule, it's a twisted ladder. The white represents the nitrogen bases that are bonded by hydrogen bonds, and the blue and the red represent the sugar and phosphate side rails of the DNA ladder. So we have an enzyme here that is unwinding the DNA ladder, and that's helicase. So it's unwinding that ladder, and it's going to expose those nitrogen bases. So then another enzyme comes in, that's DNA polymerase, and it's going to drop nitrogen bases that match each side of the DNA ladder. The other side, the same thing is going to happen with a little bit of a different process, which is beyond the scope of our discussion. But we have these enzymes acting to copy each side of the DNA ladder. So when we're done, we essentially have two exact copies of the original copy of DNA. So the process of DNA replication occurs in the nucleus. We're making another copy of DNA when cells are growing or dividing. If we are working towards a protein, and that's the job of DNA, is to make protein, we undergo another process which is called transcription. And that process involves unwinding the DNA ladder and making a copy of messenger RNA. So if we look at messenger RNA, this is shown here. It looks just like a DNA molecule. It has the sugar phosphate backbone here, which would be the gray along the side, and then it has different nitrogen bases along the middle. Except the nitrogen bases in messenger RNA are, again, they are cytosine, guanine, adenine, but instead of thymine, there's uracil. So if we look at what bonds to adenine, instead of thymine as in DNA, we see uracil bonding in RNA. So again, cytosine and guanine always go together. Thymine and adenine in DNA always go together, but if we're making messenger RNA, then uracil bonds with adenine, not thymine, because RNA does not have thymine, it has uracil instead. So then once we make a copy of DNA and convert it to messenger RNA, that messenger RNA will leave the nucleus and go out into the cytoplasm and bond with a ribosome. That's what's shown in purple here, is a ribosome out in the cytoplasm. And this is messenger RNA that contains the code copied from DNA in the nucleus. Then we have these special molecules called transfer RNA. They also have nitrogen bases, sugar and phosphate backbones, and they will come in and bond to the messenger RNA by base pairing, which means cytosine will go with guanine and adenine will go with uracil. And that will find a spot that matches based on the the anti-codon here present on the transfer RNA. That's the set of 3 nitrogen bases that matches the codon, which is a set of 3 nitrogen bases on messenger RNA, to base pair. And the little circles that we see here attached to a transfer RNA molecule, those are amino acids. And these amino acids are floating freely in the cytoplasm, but they are bonded together on the ribosome to form a protein. And a protein is formed when the transfer RNA drops the amino acid after it bonds with the correct codon on messenger RNA. It drops that amino acid, and a peptide bond forms between those amino acids. And as more and more amino acids are added to the growing protein, it eventually breaks off and becomes a functional protein. So this protein here might be hemoglobin, it might be collagen, it might be keratin, it might be insulin, any molecule found in the body that is a protein. And this protein has a very specific shape, but we'll talk more about that in advanced A&P. So the process, summing up the process, in the nucleus is where we find DNA. Messenger RNA travels through the messenger RNA travels from the nucleus to the cytoplasm and binds with the ribosome. So messenger RNA, again, is formed in the process of transcription, when we're taking the DNA strand, unwinding it, and making a single strand of messenger RNA. And again, that occurs by base pairing. Then the messenger RNA travels out to the cytoplasm, binds with the ribosome, and transfer RNA molecules by base pairing connect to the messenger RNA strand, bringing amino acids and binding amino acids in the process. Now this is really, this whole process going from DNA to proteins here, which is what we see here, the little circles bonded together, is really best seen in a visual way. So I'm going to show you a video of this process. So what we see here is a DNA molecule. It's in the nucleus, and when the cell is signaled to make a protein, we're not going to go into the process of what signals protein synthesis, but when it comes time to make a protein, the enzyme that we just saw acting to unwind this DNA molecule, remember, was helicase. So here is the enzyme shown in kind of a globular structure. That's a protein, that green worm-looking structure. That's helicase. It's unwinding the DNA molecule, exposing, again, remember, we have sugar, phosphate, sugar, phosphate. So this is deoxyribose, phosphate, sugar, deoxyribose, phosphate, and these are the 4 nitrogen bases. So if this were adenine, the other side here must be thymine, if this is the DNA molecule. If this is cytosine, this must be guanine here then. Okay, so remember that only adenine and thymine can go together, and only cytosine and guanine can go together. So you can notice that wherever we see green, we're going to see brown, because that's the only combination based on their molecular structure. So now we see some nucleotides coming in. These are little bits and pieces of nucleic acids, whether it be RNA or DNA. So again, here's the phosphate, here's the sugar, here's a nitrogen base. So if we're going to build a messenger RNA strand, base pairing occurs to build this molecule by dropping the right nitrogen base on the parent strand of the DNA. So if this is, again, if this is adenine, instead of thymine, if this is building messenger RNA, this would be uracil. Because remember, messenger RNA does not have thymine. So here's the process, building a messenger RNA. So we're simply exposing one side of the DNA molecule and building a messenger RNA strand, which is a single-stranded molecule. This process that we see occurring right here is called transcription, because we're making a messenger RNA molecule. If we were making a DNA, another copy of DNA, and we had both sides being copied, again, that would be DNA replication. But notice here we're making one single strand, which is RNA. So this is called transcription, this process we see right here. So eventually that will break off. So there's our new messenger RNA strand. In the nucleus, it's going to leave the nucleus and travel out to the cytoplasm now. And out in the cytoplasm, that newly made messenger RNA is going to bind with the ribosome. There's 2 subunits to a ribosome. So here's one subunit. This big globular structure is a ribosome. There's our messenger RNA strand. Now the next part comes in, and this is transfer RNA, or tRNA. And it's coming in, it's this looped looking structure. Again, we see the same structure, sugar, phosphate, nitrogen bases. Again, notice that the base pairing is occurring. We have this nitrogen base with this nitrogen base. So here's our adenine and our uracil, our cytosine and our guanine. Again, guanine, cytosine. Here comes the adenine with the uracil, and the colors don't matter, just as long as you understand adenine and uracil always go together, cytosine, guanine always go together if we're talking about RNA. So again, this is our messenger RNA, and this is our transfer RNA that's floating out in the cytoplasm that has an amino acid attached to it. So we'll take a look at this in a little more detail here. So here's the transfer RNA, here's another part of the ribosome. This is the second part of the ribosome that comes in. So this is where the actual process of translation occurs now. That's where we're going to bond the transfer RNA molecules that are coming in here into the ribosome, and they're binding with the messenger RNA. And with them, again, attached to each transfer RNA molecule is an amino acid. Now a peptide bond just formed between those two amino acids, and we are now building our protein. So that's the goal, again, of this whole process, is to make a protein. So that transfer RNA just left, the one that was attached to that amino acid. And now we move down the messenger RNA strand. Here comes another transfer RNA with an amino acid attached. It's going to bind with the correct nitrogen base. Again, this process we're watching here is called translation, where transfer RNA and messenger RNA are binding together with their nitrogen bases, building that protein. So see the amino acids that were attached to those transfer RNAs? They're slowly bonding together to form a protein. So this here is a protein made up of individual amino acids. Again, here's the ribosomes with the transfer RNA and messenger RNA strand, all functioning together to build a protein. So I recommend watching this video again to help you kind of reinforce this process. There's a lot of detail there, but overall, we're looking at this process. We're unwinding DNA, making a copy of messenger RNA. So when we're going from DNA to messenger RNA, we call that transcription. That occurs in the nucleus. Out in the cytoplasm, when that messenger RNA bonds to the ribosome, and we have transfer RNA with an amino acid, that base pairing is called translation. So we're taking the language of amino acids and complementing that with the messenger RNA strand. And again, slowly but surely, when we form peptide bonds between these amino acids, we're forming a protein. And again, that protein could be insulin, it could be an enzyme, it could be a structural protein such as collagen and keratin, which we find in our hair and skin, or any other protein that sustains life. I want now to look at the worksheet that I put out on Blackboard for protein synthesis. The page numbers are pretty close. It does start on page 99 and goes over pages 109. There's an excellent diagram of this process in your textbook on page 106 to 107. I'll just add that here. Great diagram on page 106 to 107. Highly recommend that. So take some time to work through this worksheet, and I will go over the answers to that. You'll see an answer key to this posted on Blackboard. So make sure that you go over this first and then check your answers on Blackboard later.